Hi, this is Miss Litton, and this is 7th period support for AP Bio review of Unit 2. Say hi.
Hi.
Okay, so if we look over the topics for this unit, 5, membrane structure and function, and then 6, 7, and 8 are all pretty much linked together. 6 was talking about energy and enzymes, how energy flows through the system, and then the importance of enzymes, because we need to know about those in order to discuss both photosynthesis and cellular respiration. I'm going to reiterate, you already have reviews set up for each of these chapters. We want to use this time to make sure we can talk about big ticket items. Now, things that come to my mind, things that I would look when I'm writing a test or looking for questions for a test on any one of these things, when I think about Chapter 5, membrane structure and function, kind of guess, what do you think might be important to me with Chapter 5? What? Okay, so to me that sounds like the structure of a membrane. Do you agree? Right off the bat, I would want to know the structure of a membrane. Because a membrane is kind of cool because a membrane provides a barrier, but yet it allows things to flow through it. Right? So that would be an interesting essay question. How does a membrane act both as a barrier and as a method to have something to get in and out of a cell, to allow transport across it? So if you're going to talk about it, you have to first by talking about all the components of that cell membrane. So what kind of things do we have? And I heard some of you say that, and that would be a phospholipid what? Bilayer. So this part right here, oh, whoops, that would be wrong. This is the part that you're really going to talk about the barrier aspect of it. Because that phospholipid bilayer, okay, has hydrophobic and what? Hydrophilic. You're like, I'm ready. Oh, whoops, I just got green. I became green. Okay, so you have hydrophobic regions and hydrophilic regions. Where are the hydrophilic regions? The outside, yeah. These are the hydrophilic regions. They like water. And then the inside is hydro what? Phobic. Okay, so it does act like a barrier. Things that are large or charged, they can't get through. Large because there's not enough space, and charged because really it's that hydrophobic region that acts as the barrier, not the hydrophilic region, yeah?
Yes.
Okay, so then we go, okay, I've drawn something else on here. What else have I drawn on here?
Protein.
A protein. Good. And then there are other components. What are some other components here on our membrane?
Steroids. Cholesterol.
Okay. All right, so we have cholesterol. And we know how cholesterol stabilizes this membrane due to fluidity, right? Whether it's hot, it keeps it all together. If it's cold, it prevents it from what? Freezing. What else do I need?
Glycoprotein.
Okay. So if I have a carbohydrate side chain attached to a protein, it's a glycoprotein. If it's on a lipid, it's a glycolipid. Yes? And this helps with cell what? Recognition. And also cell junctions too, right? You have part of that extracellular matrix that it would be a part of. All right, so that would be the structure. When I talk about the function, it is to be a barrier, but also it allows things to pass. So on function, it's both a barrier and it allows for transport in and out of the cell. So we've talked about the barrier part. Let's talk about the transport part. What are all of our options as far as transport goes?
Channels.
Straight
Through the phospholipid bilayer. Channels. Carriers. Or whole membrane. Yeah, whole membrane. So you could talk about each of those topics as well. Remember, if you're going to talk about through the phospholipid bilayer, the things that can go through are things that are very small. Name some things that can go through. Water. What else? I heard it. Oxygen or carbon dioxide. Okay, oxygen, carbon dioxide, water. Oh, those sound like the 3 important inorganic molecules, right? Yes, sir.
Like, would you say water technically can go through? But I thought it
I would say water absolutely can go through. What facilitates the process is what you're bringing up, which is the aquaporin. Am I right? Okay. So when you're going to talk about water moving through, now we're going to talk about osmosis, which is the diffusion of water across a semi-permeable membrane. Diffusion going from a higher concentration to a lower concentration. And it's here that I would talk about things like hypertonic, hypotonic, and isotonic. And remember, in all of those scenarios, right, it's one related to the other. If side A is hypertonic, then side B has to be hypotonic, because they are defined by their neighbor, right? They're defined by their neighbor solution that is separated by a cell membrane. You can't have one side be hypertonic and the other side be hypertonic, okay? You are one or the other. The only thing that's same on either side is what? Isotonic. And water must flow from the hypo. It's going to flow from the hypotonic solution into the hypertonic solution in order to maintain what? Homeostasis. Who is under greater potential danger? An animal cell or a plant cell? Why is that?
Plant cells have to turn the cell wall.
Yeah, they don't have plant cells have a cell wall to push back in case they are put in what type of solution? Hypotonic. If you are put in a hypotonic solution, water must flow from the hypo. If you're in a hypotonic solution, water will flow from the outside of the cell to the inside. If too much flows inside, you could burst. A plant doesn't have to worry about that because they have a cell wall which can push back. I could predict how a plant might function if I know its water potential. Its water potential is a factor of pressure potential and its solute potential. I smell an equation. Are you remembering the equation? Remember this? Right? And water potential of distilled water, okay, of double distilled water is always equal to 0. As you add solutes in, it will always go what? Negative. It always goes negative. It always decreases the water potential as you add items in. Water will travel from a higher water potential to a lower water potential. Okay? So if I want to calculate this out, if I don't know this right here, okay, and I want to solve for it, okay, it's like I've got A equals B plus C, right? I need to get rid of one of my unknowns. Which one can I get rid of?
Pressure potential is not a plant cell?
Yeah, I can get rid of pressure potential by finding the solution at where the pressure is equal, where no more water flows in than flows out. How could I possibly do that? Right? I could take my unknown that I'm trying to figure out and I could put it into different molarities of solution, right? And I could find that solution where it doesn't gain or lose. And I could plot that out with this being 0. And at the point where it crosses 0, that would be the molar concentration where it doesn't gain or lose. I can use this molar concentration to calculate, negative ICRT. I can use that in order to find out the solute potential, and if the pressure potential is 0, then whatever I calculate here is the water potential. Yep. Turn towards your bio buddy, see if they're in the Shadowlands, like any of that way. Got to check this group's gel roller.
Because unless it's because it's so fresh.
No, solid
If you watch,
All right, you're welcome. Okay, are we good on that? Okay, so that brings us through the phospholipid bilayer. That's one way to cross.
Yes.
And then we talked about some things related to that. Okay, then the second way we could do it is using a what?
Channel.
Yeah. It's important for Matt to yell that out. Matt, I just want to tell you, I talked about you in fourth period. Okay, or the third period. So let's talk about all the things that proteins can do in a membrane, because that's part of a cell membrane. It can be a channel, carrier, cell recognition, receptor, enzymatic, and then cell junction. Did I not teach you this?
You did.
Oh, okay, all right. So those are all the purposes. So relative to what we're discussing right now is the channel or the carrier. Okay. When you use a protein to go with the gradient, what kind of transport is that called? All right, it's passive because it doesn't require any energy. I agree with you there. What else could you tell me? It starts with an F.
Facilitated.
Facilitated transport because you're using a protein to do your diffusion with. Okay? Now, if I go against my gradient, that's going to cost me some ATP. So what do we call that? Active transport when we do that. Okay? So that is, how did your gels go?
Pretty good. A couple of my groups went through, and then we didn't get any.
Oh, okay, gotcha.
Snail that.
Perfect. You're like on top of it. Okay. You know.
Maybe.
Okay, come back to me. I'm trying to stay dialed in. Okay, so, then the third way to get across is using whole membrane. So we're thinking endo and exocytosis. For endocytosis, we have 3 types of endocytosis. Cell drinking is penocytosis. I know how you remembered it. Cell eating is phagocytosis. Stop. And then what is the third way? Receptor-mediated endocytosis, where it binds to a receptor before it does endocytosis. All right, we did passive, we did active, hyper. I think we covered it. Okay, let me just glance to see if there's anything else I'm concerned about.
Dave, that just.
Yes.
Come on, man.
He's my brother.
Okay, moving on. So, topic number 2, okay, energy and enzymes. For this one, we want to talk about types of energy. We have kinetic energy, which is energy of
Movement.
yeah, motion. Good. Movement or motion. Good. What's another type of energy?
Thermal.
Oh, thermal is heat. Good. All right. But one we talked about was potential energy, right? And specifically, you could have chemical energy in bonds. Okay. We had 2 laws of thermodynamics. Energy cannot be. It can only change forms, and every time you change forms, you lose some as heat. Overall, are we going to greater order or greater disorder? Disorder. What do we call the measure of disorder? Entropy. We eat and we breathe oxygen in order to what? Generate ATP so we can fight entropy. Right? This is why we do cellular respiration, so we can fight entropy, because we're going to greater and greater disorder. To do cellular respiration, we need a source of potential energy, right? So what do we use? Glucose, right? Cellular respiration, we use glucose. We're all together. But we can't make our own glucose because we're not autotrophs. So we are dependent on plants to do what process? Photosynthesis. So we need plants to do photosynthesis to generate sugars for us so we can do cellular respiration so we can fight entropy. But in order for us to do the best cellular respiration, we also need a source of oxygen, right? Because we need something to get reduced, the final electron acceptor, so we can keep our our electron transport chain, so we can do chemiosmotic gradient. We can form one of those so we can make ATP off of that. Yes? Yeah. So plants are also our source of oxygen. If we didn't have oxygen, we wouldn't do aerobic respiration. What would we do? Anaerobic respiration. Okay? So when we talk about Chapter 6, when we talk about the first and second laws of thermodynamics, when we talk about energy changing forms, okay, if it requires a chemical reaction, it's probably going to need to be facilitated by an enzyme, okay? Which an enzyme is a good example of one of our 4 important organic molecules, which would be a protein, okay? So we want to be able to use that enzyme to facilitate that reaction. How does it do it? It brings the reactants together on the, what part of it? Active site. It can be a catabolic or anabolic process. It can be breaking or it can be building. And I know that enzymes are very what? Specific. And what makes them specific is their active site, right? Because whatever molecule you're going to put in there, whether it's PJL or glucose, it fits exactly into that spot, right? In order to facilitate that reaction. So therefore, you can regulate an enzyme a couple of different ways, right? What are some ways we can regulate an enzyme and its functionality? We can have inhibitors of some sort. Okay, so let's talk about that. There were 2 big categories we talked about. Non-competitive and competitive. What does a competitive inhibitor do? Binds to the active site, whereas a non-competitive inhibitor binds to the
allosteric,
which ultimately changes the active site. Okay? Good. I also heard temperature. Why is temperature a regulator? You can denature it if it gets too hot, but all enzymes work at a particular temperature, right? Where the kinetic energy of that molecule is just right for it to assume its shape, right? What else could we use? pH. Because pH is a measure of hydrogen ions. The more hydrogen ions I have in a solution, those hydrogen ions can bind on to charged, right, areas, negatively charged areas of our protein. Remember, our protein's just made out of amino acids. So if you have a polar molecule, it can be influenced by those hydrogen ions. It could change its shape allosterically, right? So that would be another way to regulate it. What else?
pH.
We just said that. What else? We can activate it with a phosphate group. Kinases, right, could activate it. You might need a coenzyme or a cofactor. A cofactor would be metal. An organic structure like a vitamin could be a coenzyme that regulate enzyme activity, which makes me think about another lab. What is that lab?
Number 2.
We took hydrogen peroxide. What did we add to that hydrogen peroxide? We added catalase. Catalase, which is an enzyme which would convert that hydrogen peroxide to what? Which, oh, by the way, looks just like hydrogen peroxide, plus what? Oxygen. The oxygen would look like what? Bubbles. But we took and did this in a chamber, and we had a sensor right here that would then send it to the iPad, and it measured how much oxygen we generated in this chamber, right? So the more oxygen generated, we're assuming then the better that enzyme was able to work. And then we probably put this under different scenarios. Some of you varied the amount of hydrogen peroxide. If you would assume that the more substrate you have, the more product you would get, right? Or if you varied the temperature, you could vary the functionality. Or you varied how much enzyme you had. That could vary how much product you have. Are you all right on that? Okay. Which then, let's go ahead and mosey on to, let me make sure I talked about everything here. Oh, energy of activation. I didn't use those words. Enzymes lower the energy of activation, that hurdle. Good. Okay. Good. We yeah, they don't change the end results. Good job. Okay. So now that brings us to photosynthesis and cellular respiration. Okay. So on photosynthesis, there's a couple of assumptions we're making, and one of those is that we have the ability to capture light energy coming from the sun. Because the sun is going to greater and greater entropy, right? Which we take advantage of. Because plants can capture, as the sun is breaking down, plants can capture that energy, right? What do they use to capture that energy?
Leaves.
Be specific. What do they use? Pigments. Right? What are some pigments you know that can capture light energy? Chlorophyll A, chlorophyll B, carotenoids, right? All of those can capture light energy. And when I'm thinking about a chloroplast, I know that there are stacks of membranes. What are those stacks called? Thylakoid membranes. And that's the, what area? What do you call this? The grana. And what's the area? Other area? Stroma. Right. What happens on the grana? What? I'm waiting. What happens? The light reactions occur on the thylakoid membranes, right? And you take water and you split water and you get oxygen and you get electrons. Those sweet. Those electrons get excited by the sun's energy. They na-na-na-na-na-na-na-na-na. And then we do what? PQ. And PQ will bring hydrogen ions to the interior of the lumen of the thylakoid membranes, right? By concentrating those hydrogen ions inside, what do they want to do?
Get out.
Go back out, and we can make some what?
ATP.
ATP at the ATP synthase complex, right? Because we've established a chemiosmotic gradient.
Boom. Yes.
All right. So that same set of electrons can get excited again, and they're going to na-na-na-na. But this time we're going to use them to reduce what? NADP.
Which is NADP.
Which is what?
Isn't it NADP?
Or NADP reduced. Same idea. Okay? So as a result, when I walk away from the light reaction, I have ATP and reduced NADP that I can take to the light independent reaction that's occurring in the stroma. Okay? And there, ultimately, what I'm trying to do is take CO2 and fix it in order to get G3P, or another name for it is PGAL. In order to do that, I need a starting component. Who do I use? RUBP. Good. And RUBP, remember, is a 5-carbon unit, right? And we hook CO2 up to it, which is a what?
1 carbon unit.
1 carbon unit. And 5 plus 1 is 6. We break those in half, right? And then we give them some energy from ATP. We give them some electrons from NADP. And basically then they're fired up in their PGAL. Each time we turn this turn, we only take 1 away, so we leave some in there that can be rearranged back into RUBP, yes?
Yes.
I simplified the song. Do you want to know the simplified song?
Oh, yes.
It's up on YouTube, on this part. 3C5. I go 3C. How did I? Oh, I simplified it. Shoot, I probably better watch me.
I think we're checking on YouTube channel.
Oh, I just like this. 3C5 plus 3. Oh, dang it! I gotta watch it again. 3C6, break down to 6C3. Do you remember this? Use some ATP, use some reduced NADP on your 6C3. Take one away, PJL, Jasmine. And you're left with 5. 3C5 plus 3C1 mix 3C6, break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take one away. Now we have what? Glucose, jazz hands. And you're left with 5. Use some ATP to build 3C5 plus 3C1 mix 3C6, break down to 6C3. It's a little bit easier. But anyway, I simplified it just a little bit, okay? So now that we have this glucose, this PGAL, you can remember this is the starting point for plants. They can use that glucose to build maltose, starch. They can use it to build amino acids. They can use it to build oils, right? Whatever it is that they need. Then we take that from the plants and we do cellular respiration. And the first step is called what? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm, right? So then we can go through the steps of cellular respiration, and the point of that is to take that potential energy in the plants and convert it into ATP, the cellular currency, right, that we need to do our reactions. And we talked about what can you use ATP for. Do you remember we talked about chemical work, transport work, mechanical work, okay, and that kind of stitches it all together. Okay, so those are all of those. Aerobic respiration versus anaerobic respiration. Which one generates more ATP? Aerobic respiration. It can get you 36 to 38, maybe 32 as we're looking at it now, but you're in the 30s. Whereas anaerobic respiration only gets you 2. You get the 2 ATP that you made from glycolysis. Okay, do you want to go through all the songs?
Yes, please.
Okay, so let's go through all the songs. Let's start with water. Right? Do I want to start with water?
Yeah.
No, we'll start with cellular. No, let's start with water. Here we go. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweet, sweet. This is photosystem 2, right? That's why we're going like this, and we have 2 electrons, right? Here comes what? Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Na-na-na-na-na-na-na-na. PQ, and you make some ATP. Due to our chemiosmotic gradient. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Na-na-na-na-na. Who catches it? NADP, forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light independent reaction. Remind me to talk about RUBP's HOEDness, right? I didn't mention that. You know what I mean. Here we go.
Adjective.
Here we go. You can do it the way I taught you. I'm just doing the simple way if that helps you. 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take one away, PJL. And you're left with 5. Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take one away, glucose. And you're left with 5. Use some ATP to build 3C5 plus stop. Okay, so now let's pick up our glucose. What are we doing? Cellular respiration. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What do you get? 10. 10 of my glucose. That was awesome. Okay, 6. What are we going to do? Break it in half. How many? Here you go. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Did I really only make 2? No, I made 4, but it took me 2 to get 5 started. Okay? Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition. Right. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. Ready? 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Reductase, reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches electrons at the bottom? Oxygen forming water. But if we didn't have any oxygen, 2, oh no, we don't have any oxygen. What should we do? Fermentation. How many choices do we have? 2. You and I make lactic acid. It makes our muscles sore. Eventually, okay? Plants and yeast make alcohol. Either way, you oxidize your NAD. That's the point of fermentation, is to oxidize NAD. Okay, let me jump back to plants and RUBP. RUBP can become tempted by the fruit of another. RUBP is supposed to hook up with who? CO2. But if CO2 levels are low because stomata are closed because you're losing too much water, you're still doing photosynthesis and you're generating a lot of oxygen and you're using up your supply of CO2. So now all of a sudden, RUBP is wanting to hook up with who? Oxygen. That's bad, and what's that called? Photorespiration. So there are 2 strategies we've talked about to avoid it. We talked about C4 plants, and they hide their dark reaction or their light independent reaction in the bundle sheath cells near the xylem, the vein of the leaves, and have mesophyll cells surrounded. Remember that Krantz anatomy, that looking like structure? And that way PEP, a molecule, will fix CO2 and escort it into the RUBP. Sounding familiar at all?
C.
Okay.
C.
Another way to do it, that's a divide in space, right? But you can also have a division in time. And that would be if you are a CAM plant and you fix your CO2 at night with PEP and store up a bunch of fixed CO2 and then slowly release it during the daytime when you're doing the light reaction. Did we do a lab on photosynthesis? Yeah. What did we use?
I thought that was an actual question.
What did we use in our lab on photosynthesis? You so know. Tell me. Come on, Popeye. Spinach. Oh, I'm sorry, I didn't hear you. Yes, you used a hole punch to cut out little,
What were those called?
discs, right? Remember those little discs that you cut out?
Oh, we've just Jeremy did a terrible job.
And then you took those discs and you put them in a syringe with a solution of baking soda and you created a vacuum. Remember that? It was really super fun.
Sunk.
And they sunk because the interior of the leaf got filled with, with that water. Which we learned something. You can have too much sodium bicarbonate. You can have too much baking soda in there, remember, because it made them really heavy, right? Okay, and we put them in beakers, right? And we would shine a light on them, and we assumed the more photosynthesis they did, the more what gas would they produce inside their leaves? CO2, which would make those leaf discs rise. And after a given amount of time, we would say the hypothesis we had is maybe if you had more light, they would do more photosynthesis and rise quickly. If you had a more source of carbon dioxide, you would do more photosynthesis and rise quickly. So we could mess with all of that, right? We could have had different light that was shown on those. Those are all variables that we could do. Different temperatures, we could have cooked the leaves a little bit. See, maybe they wouldn't do photosynthesis. Those were all options. We just struggled with the whole fact of making them sink, right? But we did it. We did it. Okay? More or less. A little less than more. Okay. All right. Any other questions on this topic? Are you glad you came? Yes. No, Max. No, go ahead.
Well, I'm not sure if you'll answer this actually.
But should we be able to like design or remember
Yes.
the outcomes of these labs?
Yes, you should.
Oh.
Yeah. At any point, they could ask you to design a quantitative lab. What do you hear in quantitative? Quantity, yeah. A lab that you could gather numerical data on in order to measure some phenomena. So your phenomena you might be wanting to measure now is what?
Spinach.
The rate of photosynthesis.
Spinach.
Yeah. Okay, what else? Yes.
Can you do a quick overview of exergonic and endergonic?
Sure. So exergonic reactions require energy. Okay. If my energy starts here with my reactants, okay, if my reactants are here and my products are here, that means energy-wise, I have increased the amount of energy they have, so that must be a what kind of reaction? Endergonic. Okay, so energy had to go into this in order for it to have the products to have more energy. Where could that have come from? A catabolic reaction, an exergonic reaction. If I have an exergonic reaction that is releasing energy, I can couple that to a reaction that requires energy. That's a coupled reaction. One that releases exergonic, one that requires energy is endergonic. What else? Did that answer your question, sweetheart?
Yeah.
Okay, what else? Okay, yes?
So we know, I haven't looked at exactly yet, but I know like the amount of ATP produced per thing, like it's like 30 at this stage.
Yeah, it's 30, but you know what? It's really changed recently over the years. So the big thing I would want to walk away with, I don't know that they're not going to hold you to a number that was probably in textbooks for years now because it's varying. So what you want to see is that in glycolysis, you make very little, only 2. A large multicellular organism like us can't live on that. So we need to do aerobic respiration, and the bulk of that is made as a result of the chemiosmotic gradient on the cristae of the mitochondria, right? And so it's an oxidative phosphorylation, and that we have to have oxygen in order to do that process in the mighty mitochondria because it's the final electron acceptor. As long as the electrons are moving along that membrane, we can bring hydrogen ions and we can put them in the intermembrane space, reductase cytochrome oxidase, and that's where we're going to generate the bulk of our ATP. In order to do that, we need to have a source of reduced NAD because it's the NAD that's dropping the electrons off at the top of the chain. So the bulk of our ATP is made during the electron transport chain. You know, and you're in the 30s, you know, overall. What else? Is that it? Okay. Have good evenings. Good job. I'm proud of you. Thank you. Yeah. Oh, not the collagen cycle. You're talking about that time. No, you're talking about Do you feel smart yet? You should feel smart. You're welcome, sweetheart.